distribution of organisms.
Just one solution.
What's the basic question when we're talking about distribution? Why is it here but
Not there.
not there? That's a simple thing to ask, but it's often very difficult to answer. So if it's here because involved here, or
It moved.
just quiet up, guys. Involved here, or
It dispersed. It moved.
dispersed here. And then the contrary is true as well. If it's not here, well, why not? It didn't evolve here, or never migrated here. Or it was once here, but
It left.
it's extinct. All right. So we start talking about the largest levels that an ecologist can typically examine in terms of distribution. So the Earth is divided into 6 of these. What are they referred to as? Biogeographic regions or realms, right? And so where does one biogeographic region or realm transition into another one? How do you know you're going from one to another?
Species composition.
The species composition does what?
Changes.
It changes over
Short distance.
short spatial scales. Okay, short distance, short spatial scales. Good. Well, when we look at the planet and distribution of organisms, it's predominantly set by climate. Define climate for me again. Long-term average of what?
Weather.
Weather. Weather is short-term atmospheric phenomena. Of the attributes of weather that form climate, what are the 2 primary attributes that determine the climate? Precipitation and temperature. That's what strongly influences the distribution of organisms, all right? So when we think about climate, climate on the planet is caused by differential solar radiation on the globe and the spin of the planet, right? And its tilt. So when we think about how climate is generated, remember that we are rotating around our axes. It's hot when? During the day, when we're exposed to the sun. It's cold at night when we're on the opposite side. That's basic. But seasons are quite different. Seasons are due to the tilt of the Earth, right? So right now, the northern hemisphere is facing what?
The sun.
Closer to the sun. The southern hemisphere is
Away.
farther away because of the tilt of the axis like this. Here's the sun. Here we are in the northern hemisphere, and it's our summer. Right now, it's the winter in the southern hemisphere. So, June 21st, what date was that? That was the summer solstice where we were actually closest to the sun. But now we're starting to slowly rotate around, and eventually we'll be where? It'll be the autumnal equinox. Where's the sun most directly located at that point in time? Right at the equator. And then it will slide around. It will be farthest away from the sun. What will that be? The winter solstice, December. And then we're going to slide around again to spring. What will that be? The vernal equinox. And again, it will be right at the equator. So what you see is the most intense sunlight is moving up and down across the globe near the equator, from the Tropic of Cancer to the Tropic of Capricorn, and back and forth, causing rain as it goes as well. That's where the sun is most intense. And so that creates banding patterns of atmospheric change based on that irradiation. So what does hot air do? It rises up. And the hottest air is found where? At the equator. So as that air rises, what does it pull with it? Water, because the equator is mostly water, right, in terms of the ocean. As it goes to altitude, however, the atmosphere cools. That slows down the molecules, and what do? They shed all that moisture out in the form of rain. So the conditions at the equator are then what? In terms of heat and water, they are hot and wet. What kind of biome do you get? Tropical rainforest, right? Well, rising air creates what kind of pressure? Low pressure, okay? Now, where does that hot air want to go naturally because of a basic physical property? It wants to go towards the poles where the air is colder. What's that process from high to low concentration? That's just diffusion. So hot air at the equator is moving towards the poles because of basic diffusion, high concentration to low concentration. But there's pressure differentials as well because we have low pressure at the equator. That air has to be filled from someplace. So where's it coming from as it rises up? It's coming from the north and the south. It's sliding along the landmass, right, or along the ground. And then it starts to rise up. So it's being pulled from the north and the south. That causes another pressure differential. So that air starts to drop at 30 north and 30 south latitudes. As it drops, what happens to its temperature? What?
up.
It goes up because it's compressionally heating. Yes? What happens to its water though?
Goes down.
It goes down because where was it all shed?
In the crop.
At the equator. So it's hot and it's
Dry.
dry. What kind of biome do you get?
Desert.
You get a desert. The whole process starts up again at 60 north and 60 south. The air starts to rise, but it's not hot. It's what temperature?
Moderate.
It's moderate. It's cool. As it rises, however, it gets colder. It slows down. And what does it shed? Rain. So at 60 north and 60 south, what's the conditions? It is cool and wet. What kind of biome do you get? Temperate rainforest. And then it drops because of the pressure differentials again where? At the poles. And what are the conditions there? It is cold but dry because you shed all the moisture at 60 north and 60 south. So what kind of biome do you get? Polar desert. All the other biomes are just someplace in between. So what would you find in between a desert and a tropical rainforest? A savanna or a grassland, because there's moderate amounts of water and there's the heat is dissipated a little bit. Same thing, what are you going to find between the tropical, between the temperate rainforest and a polar desert? You find the tundra, which is someplace in between. So that explains the distribution of organisms on the planet. Whatever the plant life is, that's going to dictate the animal life. The plant life is primarily dictated by the climate, which is primarily dictated by water and heat. Okay? All right. It's more complicated than that because that basically is assuming that the Earth is standing still, is it? No. It spins. And as it spins, what it does is it deflects all of that air that wants to just go straight up, and it's going to straight up and north, and it's going to deflect it at a different angle. So basically, the Earth is spinning from the west to the east, and that makes the winds on the equator spin from the east to the west. And what that's going to do is cause all these rotational areas of wind around the planet. Okay? And that's going to influence water, because it pushes on water, and that's going to influence the motion of water laterally into these big circular gyres. And also, water is going to move towards the equator, just like air going from hot to cold. And as it does, it will actually get colder and more salty, and then sink out. And there's a big kind of global heat circulation as well. So the planet's very complex, right? But for this particular class, the things I want you to take away are, how are those climatic bands formed by differences in solar radiation on the planet? Why are the tropics hotter than the poles? What's the basic answer to that? Because they strike the equator at what?
Right angles.
Right angles. Where they strike the poles at? Obtuse angles. That means the heat is concentrated over what at the equator?
Small area.
Smaller area. At the poles, it's over a larger area and has to pass through deeper atmosphere because of the angle. So what can happen to that light? Disperse, scattered, absorbed, and reflected, and that's why it's cold. All right? So that explains the climatic conditions on the planet. It also explains some of the basic distribution patterns. One thing you notice, however, is even if you know all that information, it still doesn't answer that basic question of why here but not there. I can tell you where a tropical rainforest is going to be on the planet because wherever it's hot and wet, I'll find one. But does that mean that all the species are the same, place to place? No. They're radically different. It doesn't explain them. Lots of things come into play to deal with that. One of those is basically continental drift, but another one is simply evolution. And so we're going to talk about that as we move forward. So again, based on that basic question, why here but not there, I can predict large-scale events where the biomes are, but I can't predict what species you do. So I need a better resolution, a smaller tool. Remember, if I go to Australia, tropical rainforest, and I look at the leaf, will it look similar to the leaf that I look at in South America? Will it look the same? It might look identical. Will it function the same? It might. But are they the same species?
Probably.
Probably not. Okay? So that's the thing. They function, they look the same, but often they are not the same in terms of their evolutionary descent. They're just solving similar problems, so they have converged with that basic shape and function. So again, what tool would I have to get the more small-scale resolution of why it's here and not there?
Transplant.
I have a transplant experiment. In order for a transplant to be deemed successful, what must that organism do?
Survive.
Survive and reproduce for at least what?
One
generation, okay? For at least one generation. All right, if I move it and it survives and reproduces, what does that tell me about its potential range?
It's large.
It's bigger than the actual range. If it doesn't survive and reproduce, it's the same as its actual range. Its potential range is its actual range. Okay. So we started talking about why is it here but not there. One thing is maybe I just can't get there. What would that be? Barrier to dispersal. I just gave it to you that. So that barrier then is dispersal. What was an example I gave you? The kiwi is a nice example, right? We're talking about the barn too. So in this particular case, the kiwi is a nice example. It can't fly there, so even if you operate, survive, and reproduce, it just can't get there. All right, what's another reason why it's here but not there?
Habitat selection.
Yeah, what is habitat selection?
It can go there, but chooses not to.
It can go there but chooses not to, or it doesn't what? Recognize it as a viable habitat. That's why it doesn't go there. All right. I move it and it dies. What might be some of the reasons that it died?
Predation.
Yeah, maybe there's biotic interactions there that interfere with it that aren't in its actual range. So predation, competition, herbivory, disease interactions. What else? I move it, it dies. But I don't see any competition, predation, or herbivory. What else might have happened?
Physical, chemical.
Could just be chemical conditions, physical conditions. The soil's too rocky, it's too sandy, there's not enough water, it's too hot. What else?
Lack of symbionts.
Lack of symbionts. So lack of interdependent species. And what was a nice example of that?
The fungi.
The fungus in the pine tree, right? That was a nice example. All right, so we started talking about some experimental evidence to examine this barrier dispersal idea. And I gave you an example, experimental example that dealt with the barnacle, Balamus cariosus, yes? So what's the natural observation in terms of a pattern? Where do you find this thing?
North of.
You found it north of Point Conception. It's all over the Central Coast. But as soon as I swing down here to the South Coast, Ventura, Santa Barbara, it's gone. So what's our question as a scientist?
Why?
Why? What's our tool?
Transplants.
Transplant experiment. I'll move it from there down here and I'll see if it survives and it reproduces. So they did that. Did it survive and reproduce when it was moved? It did. Did I have confidence in their experimental results?
Yes.
I do. But why do I have confidence in their experimental results?
Good control.
Because they
Control.
had an adequate control and adequate replication. So I had confidence in it, right? If they would have not had a control and it died, would I have confidence in it? I have no idea why it died. Could have been just the what that caused its death. Maybe it had nothing to do with the water chemistry or predation or anything. It might have just been what? The transplant itself could have killed it. Transplants are pretty rough business. So we have to have an adequate control. All experiments are invalid. They're worthless unless they're well controlled. It's just conjecture at that point. All right, so let's move down here to survive and reproduce. What were our two hypotheses of why it wasn't here? Number one was the most basic, the conditions here suck. There was something about it, hot, too cold, too salty, too many predators, too much competition, whatever it is, abiotic conditions or biotic conditions are poor. What was the second one?
They can't get there.
They can't get there. There's some kind of barrier, though we can't see it as human beings. So in this case, it came here, it survived and reproduced. What does that tell you immediately? That what? That what's okay?
The conditions are fine.
That the abiotic and biotic conditions are fine. If it can get here, it's going to survive and reproduce. It's not a problem. So that leads us to our alternative hypothesis, which is there's a barrier. Is there a barrier? It is. The barrier is not one that we can see per se, but when we actually examine the water currents, then we can see what it is. So the water currents, remember, the adult is what? Sessile or motile? Sessile. It's food in place. But the larva? That's motile, dependent on transport via the currents to find new habitat. Do I want to settle down right where the adult is? If I'm a larva, do I want to settle right down near mom and dad?
No.
No, why not? Because there's too much competition there. I want to find new places that are open. That's why I go into the currents. The problem is, if I don't settle out on habitat, by the time I reach point conception, what happens to me? I get whipped out to open ocean and I die. Because I can only live so long in that form before I have to metamorphose into the adult form. That's it. So that's the whole take home of that experiment. So there was a barrier to dispersal. In this case, it was the currents flowing offshore at point conception. All right. I think that brings us up to speed, yes? All right. So here's another example. Southern California is full of this particular tree. Santa Barbara in particular is infested with it. It used to be all over campus as an ornamental tree, but it's been taken out everywhere. There used to be massive roads that are right down this kind of roadway right here between, you had to go towards the dorm between the library and the life sciences. There's a whole big giant row of them. As you went out here to Isla Vista, there used to be a massive row of them separating campus from the rest of Isla Vista. So most of those are gone. I still think there's some in IDs on the road there. But they used to be everywhere. Well, where's the eucalyptus from? It's from Australia. That's where it's native to. It's not native here. It's an invasive species. So how did it get here? It's not native.
Humans.
How did it get here? We brought it here. Okay? So the way it works is this. If you look at the acorn of eucalyptus, they get soggy and they sink. The only way for it to get here naturally would be to come across the ocean currents and land on the shore, or to be carried by birds. They're too heavy to be carried by birds. They get dropped, and they're too heavy and they sink, so they can't make it naturally. So the only way they can get here is artificially, and the way they get here is us. Why was it brought here in the first place? Does anybody know? What's that?
Railroad.
It was brought over here for the railroad industry. So eucalyptus grow really fast. And so the idea was you could grow this tree really quickly, chop it down, and then use it as railroad ties. But there's a big problem with that. It doesn't work. Why not? Say it again. It's not that it's too soft, because you can dry it and cure it pretty hard. It's another reason. Not that it grows at an angle, but you're very close. It grows in a way that's not conducive for cutting straight railroad ties.
Corkscrew.
It grows as a corkscrew. That's exactly it. So next time you see a big blue gum eucalyptus, right, the ones with the white bark and it's kind of scaly on the outside, peeling off, go up to it and look at it, get below it and look up. And what you'll notice is it grows like the corkscrew, like this. It looks straight, but it's actually growing like this. So that means when you try to cut straight pieces of wood out of it, what happens? They bend, they twist. Do you want to have railroad ties that are twisting? No, because then your railroad goes, your cart goes right off. It's not as good. So here's the thing. They brought it over here for that purpose. That's kind of a dumbass thing to do. Wouldn't you think you'd cut a eucalyptus tree down in Australia and see if you could make ties there before you brought it over here? So it got here, and lo and behold, they couldn't use it for that purpose, but the thing grew like a weed here. It loves it here. Why?
No predators.
Well, the first thing is it's got favorable abiotic conditions. The habitat type in terms of soil, water, and temperature are very conducive to where it grows in Australia. So therefore, it loves it. But it has another benefit when you bring it over here. What it lacks over here?
Predators.
All its predators and all its diseases are no longer here. So it grows like crazy, now competes the natural vegetation. Next time you take a look at eucalyptus, go below it and look at the ground after you look at the spiral. Now what do you see growing below eucalyptus? You ever noticed? Smaller eucalyptus, some real tough grass, and then nothing else. And the reason is eucalyptus has its own deterrents, chemical warfare agents against other plants. So what happens is when it rains or it gets foggy, water drips off the surface of the leaves and picks up toxic chemicals referred to as allelochemicals, which then fall to the ground and poison the ground against the germination of other plants except eucalyptus. Right? And so they basically poison the soils except for themselves. They're a very ingenious plant. So that's bad news for anything else here. So some of the things that do consume it, that's one of them right there. What's that?
Koala.
That's the koala bear. How many people love the koala bear? Raise your hand. Hi, don't lie. All right. Yeah, you're deceiving yourself. The koala is not all it's made up of. First thing, I hate to burst your bubble. Is it a bear? No, it's not a bear. It's a marsupial. It's called a bear. Common names are worthless. It's not a bear. It's nothing to do with bears. All right. The next thing is, are they nice? No, they're actually very mean. They're ornery things. One of the things about, there's some common urban myth about them too. So those allele chemicals are highly toxic. If you eat them, they are also highly narcotic. All right? And can cause all kinds of strange physiological problems for mammals. So the idea was, these things are awful slow, right? So what was the idea that they were so slow? They were always hot because they eat narcotic loads in the leaves and that's all they eat. That's actually false, all right? They're so slow because there's so little nutritious browse in terms of the eucalyptus. The energy return in it is so low that that's why they're so slow. There's not enough energy for them to be quick or fast. They can't eat enough of it, and it's hard to digest. That's why they're slow. Also, in terms of their intellect, they're morons. So per unit body mass, they have one of the smallest brains of mammals. They're not the brightest tool in the ship. So next time you look at the koala, I want you to look at it with a more critiquing eye, okay? But anyway, why is the eucalyptus not here then? Naturally, it does a great job because there's barriers to dispersal, which is the Pacific Ocean. It can't make it across on its own. Why is it here? Because that barrier has been breached by us. All right, so that's another example of a barrier to dispersal. You want to hear some fact that's worthless but interesting, kind of koala related?
Yes.
So, if you know where Tasmania is? So Tasmania is a big, large island off the southern coast of Australia. And Tasmania is home to some of the very largest legal opium poppy fields in the entire world for the production of methadone. What's the not heroin, but it's derivative. Not fentanyl. Morphine, thank you. Morphine. So that's where they produce morphine from. There's massive fields of it. And so they're all restricted and everything, and they're all fenced off. And so what they were finding in these giant opium fields, and not opium fields, those opium poppy fields, where you guys ever seen those like the crop circles? You ever see those where like the crops are laid down in those concentric circles and all that kind of stuff? And you know, there's all this stuff, it's aliens and all that. Well, they found these things there. First off, is it aliens who leave crop circles? There's a famous story in England where they had all these crop circles, right? And it was funny because there was all these supposed experts going out there, scientists too, and saying, well, there's no possibility this grass is laid down at perfectly right angles to itself, and there's no sign of other disturbance. There's nothing that we know that could possibly do this. It must be something that we have no idea about. Was that accurate? No. You know what they found after like 20 years of this or something like that? These two guys, these old guys, came forward, and they were these old British guys. And what they would do is every Friday they'd get drunk at the pub, and then they'd go out and they'd make crop circles. And they did it with the most low-tech thing ever. You ever seen baseball caps and they have that little button on the top? So what they do is they put a piece of string tied to the button. And one guy would stand in the middle of the crops, and the other guy would go out on a line with a piece of string with a big, long 2x4. And he'd just walk in a circle around the guy, and they'd make shapes drunk. And they did that for 20 years, and then they would laugh all the time at everybody talking about the aliens. So anyway, these things started turning up in these opium fields, right? The poppies. And so they didn't know what was going on. They couldn't figure it out. And so they put all these cameras out there because it's such a valuable commodity as a crop. And what they found was something quite unique. So in Tasmania, there's lots of wallaby. You know what wallaby are? They're like a small version of a kangaroo. They're kind of shorter and stouter. And wallaby are attracted to the smell of the opium poppies because they're really sweet and they're also edible. And so they had these big electrified fences and everything to keep animals out. But that didn't stop the wallaby because why? Because they're fabulous diggers. And so they smell all that stuff and they dig right under the fence lines to go and feed on the poppies. But then what would happen is because it's not native to Australia, it screwed up their physiology big time because they're eating the opium poppies. Right? And what would happen is it would screw up their ability to locomote properly. And the way they operate as a group, right? I can't remember what the name of their group is. It's got some funny name, clan or something. But anyway, I can't remember. It's got some interesting. Anybody remember what a kangaroo group is called? No, it's got some interesting name. But anyway, they have a leader, and they follow the leader. So the leader would get messed up physiologically because they'd be high as a kite on opium, and it causes them to either veer to the left or veer to the right. So what would happen is they would start to jump to get out of there, but they would jump like this, and they'd all follow behind. And so they'd be going around in these big circles in the plots like that and laying down the things creating crossroads. That's completely useless, but that one's actually true. Unlike the koala being high, which is. Let's talk about the rat and the mongoose. This is an interesting one. So if you go to Hawaii, especially the Big Island, the first thing you notice is there's tons of mongoose running around everywhere. Do mongoose belong in Hawaii? No, they don't belong in Hawaii. Where are they from? They're from India. That's exactly right. And so here's the thing. There's also a ton of rats there. Do European black rats belong in Hawaii? No, they belong in Europe. But there they are. And so the problem was, as European ships made their way to the Hawaiian Islands, there were rats in the holds of the ships. And as soon as they got there, they jumped off the ships and they jumped onto the island. And lo and behold, the island is full of birds. And these birds, since they don't have any natural predators or didn't, are ground-dwelling birds who made their nests on the ground. Because there's no rats or anything like that on the Hawaiian Islands naturally. So when the rats got there, they started to decimate the birds. They eat the adults, they eat the juveniles, and they attack the eggs, and they were destroying them. And they were causing mass extinctions of the birds, which they're still doing. So the idea is, well, how the hell do we get rid of the rat? They're killing all these beautiful Hawaiian birds. And so they tried trapping them and poisoning them. Rats are super smart, and they're really adaptable, and nothing worked. So the idea was, well, is there a biological control for getting rid of the rats? Is there some way we can biologically control them? So they looked around to see what would be a good predator of rats. And this is the one that they found. So when the rat was introduced, the rat populations grew at the expense of the bird populations because they consumed them. So the idea was, we'll add the mongoose, because the mongoose is a voracious predator of rat in certain areas of the world. It really loves to eat them if given the opportunity. So mongoose were brought over, started at the Big Island, and they were released to control the rats. Well, when the mongoose got there, they love the Hawaiian Islands, but it's very similar to their habitat where they come from in India. And they prospered. They did great. But there was a big problem. They were designed to get the rats, but did they? No, they did not. So what happened was, when the mongoose were introduced, what happened was they fed on the birds. And they fed on the bird eggs too. The reason was pretty interesting, actually. They would prefer to eat the rat if they could. But the problem was they never saw the rats. Do you know why? Because the rats are nocturnal, but the mongoose is what? Diurnal. So it's up and about in the day just like we are. And the rats are up and about at nighttime. So they never meet. So if there's nothing to eat in the daytime that's ratty, what will the mongoose do? It will eat the birds, which are up and about during the day, and the birds ate. So that devastated the birds even further and drove multiple bird populations to extinction. So this is a real screw-up. Again, you would think somebody would have the common sense to test this stuff before you actually unleashed it, right? To test whether this would work. But they didn't, and now we have this big nightmare. So now we have a double screw-up. Introduce rats, introduce mongoose, and you can't get rid of either of them. And that's real problematic. So in terms of distribution, we have organisms that are surviving and reproducing fine in the habitat. Why wasn't the rat there originally? It can't fly there, it can't swim there. This is all the way from Europe to Hawaii, right? It's not happening. But they got there via artificial mechanism, which was our transport. Same thing with the mongoose. It's not flying or swimming from India to Hawaii. How did it get there? We brought it there. We reached the barriers to dispersal. This can also show the highly negative impacts of invasive species in habitats. Let's talk a little bit about habitat selection. Remember, this is, I see it, but I don't recognize it. or I see it and I choose to go there or not go there, right? If that's a barrier, I'm choosing not to go there. Well, why not? Well, a lot of this has to do strictly with just the behavior of the organisms in selecting the habitat. So here's a nice example. This is the dragonfly. Dragonflies are voracious predatory insects, okay? They have a voracious predatory larval stage that operates in water, both in lakes and rivers, and then there's also the terrestrial version that you're most well acquainted with. They're fabulous flyers, much better than birds or bats or anything like that. Not only can they go forward, they can go backwards, up, down, and diagonally. So helicopters are actually modeled on a lot of the movements that dragonflies can actually produce. Many of the things that we have as technology are biomodeled on things in the real world. You want to see some crazy stuff? You ever seen a stealth B-2 bomber? You seen that crazy thing? That shape is exactly the same shape as a swallow in full flight, because it's amazing aerodynamic. You put them side by side, they're almost identical in terms of their overall side view. It's really neat to look at. All right. So in this particular case, when you go out to habitat with dragonflies, what you often see is during mating season, like if you go to a lake or a river, you will find male dragonflies defending habitats of about, depending on the species, of about a meter, about this long, okay? Sometimes bigger or smaller depending on the species. And what they do is they go around in a circle, about a meter, sometimes 2 meters, depends, but mostly around a meter, and they're zipping around in circles like that. And then right here's another one doing the same thing, and then another one, and they'll encircle a lake like that, or they'll go right up the sides of a river like that. One piece of habitat doesn't look much different to another, and potentially any dragonfly can operate all along that stretch of habitat, but they don't. Only in that particular stretch. So they're selecting a very small portion of the habitat versus the overall habitat. What's your question? Why? And so you can answer that question of why it's here. Why is this one here but not over there? Looks perfectly fine to me. There's one over there, but it's not over here. Well, the reason is because that's the area that they can potentially defend. Any bigger than they can't defend it. This is how it works. Female dragonflies fly up here when they're ready to mate. And the males are down here guarding the territory. The females go across the territory and they eyeball it. And based on the characteristics of the territory, they decide whether or not that's the best place to lay eggs that are going to generate the most success. Typically, the most fit dragonfly males are defending the best habitat types. All right? Now, here's the thing. As she comes down into the habitat like this, he will meet her, mate with her, and then she'll lay the eggs and fly off. Okay? Now, here's the thing. If this goes past a meter, and let's say it's 3 meters or 2 meters out like that, and he's making those rotational rounds, she comes down right here, but he's way out over here. Before he can come back to get her, what happens?
Another one.
The one that's closer over there, who's defending the meter territory, flies in and mates with her before he can make it back from the 2 meter distance. And he gets basically completely outcompeted for reproductive success. Does that make sense? So what that's doing is constraining them to these small areas that they can get back to at any one point in time to mate. Any larger than that, they can't. So that's why I'm only at this 1 meter and not over the 5 meter area or something like that. Does that make sense? So that's the idea of habitat selection. They choose not to do it because their reproductive success goes way down. All right. So again, behaviorally constrained distribution. Here's another example. So these are some little brown birds. Unless you're an ornithologist, it's almost impossible to tell the difference between, I can't tell the difference between them when I just look at them. So this is Anthus trivialis and Anthus pratensis. And so when we look at them, they're commonly referred to as the tree pipit and the meadow pipit. They do basically the same things. They look the same, they sound the same, they eat the same, they do everything the same except for one little thing. And so if you observe them, they have one little difference in the way that they do a mating song. They're called pipits. Why?
They sing.
Because that's how they sound when they sing. They just go pip, they make a little pip noise. A lot of organisms, the naming of them is not very imaginative, right? It's, the blue whatever, why because it's blue. And these things pip, they just go pip, and that's why they have that name, the pip. So when you watch them carefully, what you see is they actually both can breed on the ground, That's where they set up their nests. But you only find tree pipits in areas where they have isolated trees in the meadows, and then you find meadow pipits where there aren't any trees. What's your question?
Why?
Yeah, why? So when you look at it, we want to know what limits the distribution of the pipits. They're eating the same stuff, doing the same stuff, looking the same way, breeding the same way. What's going on? Well, the issue is this. When you watch their breeding song, there's one little difference in it. It sounds almost identical. You can barely tell the difference between it. But right at the end of the song, the tree pipit flies into a tree. The male tree pipit will fly into a tree, and on one of the branches will go, pip. And then the meadow pipit stays on the ground and goes, pip. That's it. That's the only difference. However, if I'm a tree pipit and I don't do my last pip in the tree, female tree pipits will not mate with me. If I pip on the ground and I'm a tree pipit, no tree pipit will mate with me, no meadow pipit will mate with me. So I don't have to get any mating at all. It's that one little weird behavior. So that means that male tree pipits are relegated to what habitat? Habitats that have trees in them. Okay? That's the whole idea of it. So a similar song, but where they end that song is quite different. So again, the take home on this then is, there's a behavioral limitation of the distribution. Tree pipits are only found where there are trees. Why? Because they can only make their mating song perfect if there's a tree, and that's the only time they have reproductive success. Anywhere outside of that, they're not going to have reproductive success. So that completely isolates the two groups, even though they look quite similar. All right, why is that maintained? Because if you do that, and you pip in the tree, you leave more descendants than once you don't. And you quickly establish a population where they all pip in the tree. All right, we're going to shift gears. And now we're going to start talking about biotic interactions that affect the distribution of organisms. So biotic interactions are basically anything like predation, herbivory, competition. And so in these particular cases, what you will see is the distributions of organisms are really limited by the presence of other organisms, both within species and between different species. So competition, predation, parasites, and diseases are really strong structuring agents for the distribution of organisms. So when we think about it, we're going to start off with competition. Competition is a negative interaction by definition. What do I mean by that? If you're involved in a competitive interaction, you are doing worse than if you were not in that interaction. And how is it measured? It's measured at population growth levels. So why is that the case? If I have to compete, I have to burn up energy that I can put towards what? Reproduction. And therefore I have less reproductive output. I can get hurt, and I burn up a lot of energy. So if that's the case, you do poorly. So it's negative. The population growth rates of organisms involved in competition, either within a species or between, go down when you're competing. All right, you can compete over anything, any kind of resource, space, light, food, water, whatever it might be, mates, anything. Anything that's in limiting quantity, you can compete for. So you can compete intraspecifically. That's competition within a species. College is based on a competitive interaction. What do you compete for? You compete for grades. You compete for mates. Those all kinds of social stuff. The nature of our world is competitive. The nature of the universe is competitive. The nature of atomic particles is competitive. Oxygen is one of the most competitive. Why do people say, why is competition so common? Because the core material of the universe is competitive. Think about oxygen. Oxygen is the most competitive molecule there is. It has an open valence structure. Remember from inorganic chem? It always wants to grab what to become stable? An electron. And it rips it off other stuff to do it. It's in full competition for them. That's why if you leave a car out on DP for a year, what will it look like?
Oxidized.
It won't just be dusty, it'll be wet. It'll be rusty because the oxygen has torn off all the electrons all over it. Why do I look way older than you? because oxygen's been beating the hell out of me for a lot longer than it has you. All right? So oxygen is a competitive molecule. That's why our universe is a competitive place. And biology is no different. So competition is quite vanilla. So competition within a species is referred to as intraspecific competition. Competition between different species is referred to as interspecific competition. And then there are forms of that competition. So two baboons warring over meat, what's that? Intra or inter? That's intra. Hyena versus lion. That's inter. Okay? Now let's take a look at the form of it. So competition occurs in a variety of forms. The first is interference competition. This is a direct physical interaction over a resource, right? Species A competes with species B over a resource physically. So the example would be just a simple example, right? There's a hamburger. We both want it, right? And so I come up and I slap you and I take the burger and you're stunned. Did I directly physically interact with you?
Yes.
Absolutely. Now you suffered because you didn't get the hamburger. But I also suffered from that interaction. Why?
You used energy.
I had to expend energy to get him, and if I didn't get out of there, maybe he'd beat my ass, right? So there's all this negative consequence. Even though I got the food, I still had to expend more than if I could have just walked up and he wasn't there, I wouldn't have expended anything. So that's why it's still negative, right? So that would be a direct physical interaction. Now let's do it again. The second form is exploitative competition. So in exploitative competition, you're talking about competition for a resource that's mediated through the resource without a physical interaction. So in this case, there's the burger again, right? But we don't touch each other. I just see it and I'm faster. I run in and I grab it and I run away. Did he suffer? He didn't get food again, so he's hurting. But did I suffer? I had to expend all that energy to be quick, run, do all that stuff. I might have fallen down and broken something. So that again, there was no physical interaction over the resource, but it was mediated through the resource, exploitative competition. So the way you code this stuff when you want to talk about it is, first you define whether the interaction is within or between a species, and then you determine what the form of it is, right? So in this particular case, wild dog is biting the butt of hyena. What would this be kind of competition would be referred to as? And they're fighting over a territory. Interspecific because they're two different species, and interference because it's direct. If I bite you in the ass, it's a direct interaction, yes? All right, let's look at the bottom one here. We have all these ungulate animals. We have wildebeest, we have the zebra, we have gazelle in there. They're all walking next to each other, but they're not touching each other. They're all eating the grass. The more of them there are, the lower the growth rates of all those populations are. What form of competition is that? That is, again, interspecific competition, but what form is it? It's interspecific exploitation competition because they're not touching or physically interacting, but if I eat grass, it's less for you to eat, and you're going to suffer because of that. So that's how you do it. What's the form? Within or between a species, and then finally, is it direct or indirect? That's how you would code it. So what kind of natural patterns in the world would indicate that competition was resulting in the distribution of organisms? Well, the first thing you can look for is if I have two organisms and they're basically side by side, if they're competing for something, if I take one of them away, what should the other do?
Take over that building.
What's that?
It should take
It
over.
should take over whatever the space was that the other one was occupying. So if they're side by side like this, right, species A and species B, and I eliminate one of the species, well then species A should occupy the whole resource gradient. That's referred to as competitive release, right? Well, that makes sense. They're released from competition. If they occupy new habitat or resources, that's also referred to as a niche shift. We'll talk about what that term means in about a lecture or two. So they're occupying new space, new habitat, new resources. So if you open up your available space where the older one used to be, that's competitive release. Well, what might that indicate then? If I go around the world and I see places where species are lined up side by side without hardly any overlap, but they're just kind of on this boundary, that's probably a pretty good indication that they're competing aggressively. That's why. If they were totally overlapped, what would that tell you?
They're not.
Yeah, they're not competing with each other because they're allowing each other to overlap, probably, all right? There's not a lot of physical an interaction there that's keeping them from that. So they're basically dividing because of too much competition. So those would be natural patterns that might indicate competition is going on. Let's look at some experimental information. So I'm going to introduce some experiments by Joseph Kahn. So Joseph Kahn is a famous aquatic ecologist for the most part, but also done some work in other areas as well, in the tropics. He is a faculty emeritus here. He's still on campus. You can go talk to him if you want. I was talking to a student the other day who's engaged with him. He's incredibly famous. He's thought as kind of the father of experimental aquatic ecology. He's done some fabulous work, and some of his ideas have basically educated us on how the world works naturally all around the world, and his work is used widely. So I'm going to introduce some of his early work today. And part of it was done in marine intertidal fields in Great Britain. And so when you go to I've been to England before. So if you've been to England, it's an interesting place. I like it quite a lot. But when you go and you take a look at the coast of England, the first thing you'll notice is the tidal swings are much greater than here. So we don't have much tidal swing. So even our lowest tides, we're only looking at like 6 foot at most, right? Well, you could look at triple, quadruple that in terms of England, in terms of the area of land that will expose or inundate. So you have these massive tracts of intertidal field that are opened up, and it's showing rocky reef like this. So this work was done in the 60s. So I'm going to be introducing some classical works in ecology to you guys to kind of form the bedrock of ecological thought, right? And so this is one of them. So this is what he found. When he looked at it, he noticed that these rocks were covered with barnacles, and they were barnacles of different sizes, which ended up being different species. And so there was a large barnacle called Balanus, and then there was a smaller barnacle called Thamylus. And so when he looked at them, he found an interesting observation. Thamylus were all up in the upper intertidal, and Balanus were all down in the lower intertidal. All right, so there was more to it than that. So here's the interesting thing about it. When he went and he watched what was happening during the breeding cycles and the movement, remember these are barnacles. So the adults are what again in their life cycle? They're sessile, they're glued in place on the rocks like a little volcano, yes? But what about the larvae? They're motile, and they wash in and all around the reef. So this is what he found. There was tight zonation of the adults. Small barnacle in the top of the intertidal, kind of large barnacles in the mid to lower intertidal. All right? But the larvae, they settle out everywhere. So again, the red arrows are indicating the distribution of the adults, and then the green arrows are introducing the distribution of the larvae. They're everywhere. So what's your question then?
Why?
If they settle out all over the rocks, both places, right, if you're big or small, why is it that the adults are only found high or low? Do you have a tool to test this? You do. What's your tool? The transplant experiment. What are you going to do? You take the big one and you move it where? Up. And you take the small one and you move it where? Down. And you see if they survive and reproduce, to answer the question of what might be limiting that distribution. All right. So the question is, why is there zonation in the first place? And what limits that distribution of the big and the small barnacle? So this is what he did. He moved them via transplant, just like we talked about. You take this big one, you move it up. You take the small one, and you move it down. So here's the first thing that happens. When you move the big one up, it dies. Any guess why? It dries out. That's exactly right. So I want you to look at them carefully. So remember, we're looking at kind of a torpedo shape, and it's facing directly towards you with an opening. And you see the two little valves where they kind of close, and there's like a little dark fissure that runs down the middle. Do you see that? I think I have my point on it. All right, here's the big barnacle. This is one barnacle right there. You see this little dark fissure? That's where the two tests of the shell are coming together and closing. But notice there's a pretty big gap right there, right? Now take a look at the small barnacle. Here's one right here. You see that dark fissure? You hardly see it, right? There's one right there, one right there, but notice that the fissure is a lot narrower. So why are they closing up like that?
It's drier.
Because why?
To conserve water. Like
Water retention. So as the intertidal goes out, they close the shell to keep the water inside so that when they're exposed to air, they don't desiccate and die. That's the whole purpose of it. Now here's the thing. Because of the morphology, which one can close tighter?
Smaller
Well,
one.
the small barnacle can close way tighter than the big barnacle. So if the big barnacle is exposed to air for too long, what happens to it? It dries out and it dies. So that limits it to lower areas of water where it's not completely exposed for too long of a time in the sun. So that's what's limiting its distribution to the mid to lower intertidal zones. So Thammus can close a lot higher than Balamus, which is the large barnacle, and that's why it's found high, but Balamus cannot be there. That explains one distribution limit, but it doesn't explain the bottom one. So why is the small one not low? So this is the experiment that he set up. So basically he marked out quadrats, which are just basically squares on the rock, all right? And then in these quadrats, he divided them in half, and then he let natural settling occur of the small and the large barnacle, because they settle all over the rock. Then what he did is he went in and he divided it in half, and on one side, he physically removed the large barnacle in the mid to lower intertidal. Okay, so this is an experiment in the mid to lower intertidal. And all this is replicated and all this is controlled. So, he comes back at X amount of time later, and this is what he finds. In the control where he didn't touch anything, he just let the barnacles settle out. The big barnacle got really big, and all of a sudden the small barnacle was eliminated from the plots. But where the big barnacle was removed physically with the experimental treatment, who's still living there and doing fine? The small barnacle. So what does that tell you about the mid-lower intertidal field conditions for abiotically for the small barnacle?
Very good.
Are they okay?
Yeah.
Yeah, they're fine. So chemically, whatever those physical features are, there's no problem at all. That little barnacle is doing great at the lower intertidal. So why is it not there?
Competition.
Well, if you look how big up Balamus got, right? Remember what the shape of these things look like. So they're like little volcanoes, right? If I'm a little barnacle, as this barnacle grows, what's it going to do? It's going to get so big that it's literally going to pop me right off the rock and lift me off. You guys have all used a chisel or seen a chisel before? That's exactly what they do. They have that kind of edge shape. You stick them in, you hit it, and you pop stuff right off. And that's exactly what they do to the small barnacle. So by growing aggressively, they grow underneath them, lift them off the rocks, and get rid of them. Why would they want to do that?
Make more living space.
Yeah, more living space, because they're competing not for food, but they're competing for space to get the food. Remember, they're filter feeders, right? So you need a place to hunker down, and space is what's important. There's plenty of food in the intertidal, but you have to have a place to grab it. So if I have small barnacles in the way, this is a way to get rid of them. I can't do it to other barnacles that are my same species because they're the same size and they're exerting the same force on me, so I can't pop them off. But I can pop off the small ones. All right, so that explains the distribution limit of the small barnacle. So Thammus survives in the lower intertidal if you remove the big one. However, its lower limit is actually set by a biotic interaction, and that's competition. The big one grows faster and it pops it off the rock. So let's review it real quick. What sets the upper limit to the distribution of the large barnacle Balamus?
Desiccation.
An abiotic factor, which is
Desiccation.
desiccation. What sets the lower limit to the small barnacle Thammus? A biotic factor, which is not just competition. What kind of competition? Interspecific interference competition. It's interspecific because
Between
it's
species.
two different species, and it's interference competition because it's direct and they touch. All right? That's it. That's a cool story, but there's more to it than that. If you go a little further down in the intertidal, you see another organism, and it's a big predatory snail called Thias. So predation is not a negative-negative interaction, it's a positive-negative interaction. Who gets the positive interaction in terms of their population growth rates?
Predator.
Yeah, the predator, they get to eat, right? They turn that into reproductive output. Who obviously suffers?
Prey.
Prey. If somebody eats you, reproductive output's not so good. If I take a bite of you, are you doing so well?
No.
No, now you're injured. What are the impacts that a predator can have on the prey? I can consume you. That's a pretty massive impact. But I can also do different things to you too. I can injure you and not kill you. but I can also modify your behavior because you're scared of me and don't want to die or don't want to get partially eaten. So what are the 3 things that you can do if you're prey to avoid being consumed?
Hide.
Let's do a thought process, okay? Lion, hungry, at the door. Us in here. That's the experiment, right? So, what are your options?
You can hide.
The first one is probably what? Run. Then run and run fast. Now again, you have to run faster than the lion?
No.
You just have to run faster than someone else in the room. Okay? That's hard rule. That's how it works, right? Run too fast than the lion, just faster than the person, behind you or whatever. So that's why you can run away. What else could you do? You can hide. Hiding can be quite effective, especially if you happen to hide somewhere in the back of the room and the lion's getting satiated on everybody in the front of the room. You might be able just to saunter right out the door and the lion wouldn't even care later on. What's another, the third way? You can fight. Fighting can work. You can also be quite unsuccessful. It really depends. So here's the lion. You're first up, my friend. What's your odds of success? You're delusional. You're in denial. You're a flaming yellow on the lion's dinner, my friend. However, if you're the guy in the black shirt in the very back of the room, and you're the last one on the pile, what's your odds of survival? You. Your odds of survival are damn good. Alright? Because all these other people have been injured and destroyed, and by the time it's to you, you just basically have to put a pinky on it, because the line's buried underneath everybody else. So it really depends. So those are the 3 things that can happen. So you can get consumed, or you get behavioral modifications. You can run away, you can hide, or you can fight. Those are your 3 options. Alright, so in terms of this particular snail, how might it impact distribution patterns in organisms? So, this is another Connell experiment. It's an intertidal predation experiment. Thias is the predator. Here it is, right over there. It's a big predatory snail. Here's Balanus, that barnacle we just talked about. So when we look at the distribution patterns of these organisms, this is what it looks like. You go to the low intertidal, and it's all snail adults. You go to the mid to low intertidal, it's all big barnacle adults. However, the larval forms of both, which are both motile, settle out all over, mid and low. Again, what's your basic question? Why zonation? What's our tool for testing it? The transplant experiment. What are we going to do with the snail? Move it up. And what are we going to do with the big barnacle? Move it down. And we're going to see what happens. All right? There's now a complexity in this, however. When I move a big barnacle down, there's no problem because it's on a rock. I move it down, can it go anywhere?
No.
Stuck there. But if I move a snail up and it doesn't like it, what can it do? It can move right back down on its own because the adult is motile. So what difference do we have to have here to keep it in place? I got to cage it or tether it or do something like that. Now that's problematic. Why? That's stressful. I'm changing the behavior of the organism. It might not like to be there. And that might cause this transplant to fail, yes? So it's kind of just, whenever you're thinking about these things, think how you feel about it, right? It'd be basically the same thing. I rip you out of your house. I grab you. I throw you down someplace else you don't want to be. And then I throw you in a cage and say, act normal. And now reproduce for me too. Is that going to happen? Probably not. So again, you really have to kind of think about the quandary you put these organisms in when you do these kinds of manipulations, because they are feeling and thinking creatures too, right? So in this particular case, we're going to use that transplant experiment to answer the basic question. Why low for the Thias and why high for the Balanus, and no overlap of the adults when the larvae settle out everywhere. We're going to use a cage here. And so that cage is going to keep the snail up where we want it. But here's the problem. This is a predator. If I just go and stick the barnacles down at the bottom, what's likely to happen to them? They might all get eaten. So what's one way I can prevent them from getting eaten?
Keep those snails out.
I'll keep the snails out by caging them out. I cage them in high to keep them there, and I cage them out by circling the barnacles that I move. And then I control for all those different things. Okay? I control for the transplant itself, and I control for the cage. Well, when you do that, this is exactly what happens. So the first thing is, in the cages, when I move the big barnacles down, they survive fine. What does that tell you immediately about the abiotic conditions of the lower intertidal field for the big barnacle?
They're fine.
They are fine. They do well there. Okay? So what's limiting their distribution then? Biotic interaction, which is predation. If you move them down, the snail will consume them. That's why they're not there. All right. Now, why is that snail not up in the mid-intertidal? Because that's where all the good food's at. What do you think happens there? If I move it up, what happens? It dries out. That's exactly right, and it desiccates. Now, here's the thing. When you look at the snail and I flip it over, it has a big opening where the foot has to come out, right? And even though they can retract that back into the shell, it's still a lot of surface area. And what happens if they're exposed to air for too long, they will desiccate, they will die, they'll suffocate. So what you see these snails doing are they make runs on the tides. So as the tide goes up, they follow it up, they feed, and then they get the hell out of there before the tide drops. And if they make a mistake and they go too far up, what happens? They're dead. All right? So it's kind of a game of chicken with the tides as they move up and down and how fast you are as a snail. So that's what limits their distribution. If they go up too far, they desiccate and they die. So that's a biotic interaction, abiotic interaction of desiccation. So again, lower intertidal, that one's going to be set by predation for the barnacle. Mid-intertidal separation for the snail is set by abiotic desiccation. So let's run through them all. Now we have tight zonation patterns. What does it look like? Small barnacle at the top. Big barnacle where? In the middle. And who's at the bottom? The snail. What sets the upper limit of the big barnacle balance?
Desiccation.
Be more precise. An abiotic factor, which is desiccation. What limits the small barnacle's lower distribution limit? Biotic interaction, which is? What did I just say? I meant the small barnacle. What limits the lower distribution of the small barnacle? A biotic interaction, which is? Interspecific. Interference competition. Very good. What limits the upper distribution of the predatory snail Thias? Abiotic factor, which is desiccation. What's the lower limit of distribution for the big barnacle Balanus? Biotic interaction, which is predation. There you go. So you can see how abiotic and biotic interactions interact with each other, right? To control the distribution of organisms. You can even go lower, and there's another level. If I go lower, I see that the snail gets eradicated by algae of all things, right? And there's a whole series of interactions that are responsible for that. But for our purposes, this is where we're going to end that particular story. So that's a nice example of how biology interacts with the physical and chemical environment to dictate distribution. So let's spend some time talking about the physical and chemical environment, abiotic factors. Of those two things, temperature and water are always paramount in terms of controlling distributions of organisms. So when you look around the planet, what you see is organisms typically have an upper lethal limit of temperature and a lower lethal limit of temperature. And you can solve that in a variety of ways. You can get pretty close to those boundaries. Some have really large ranges, others have much tighter ranges of tolerance. So, for instance, when dealing with temperature, the range is typically 0 on the low end, Celsius, and then 45 degrees on the top end, Celsius. So, why is that? What happens to cells at 0 degrees? They freeze. What happens at that point? What happens to the cells?
They burst.
They burst. Why do cells burst at 0?
They're mostly water.
Because cells are full of what?
Water.
Water. And at 0, water turns from a liquid to a solid. And what happens is it has its conformational shape change to align its bonds properly, and it creates more area than does liquid water where the molecules are moving in and out like this, right? So it's all rigid, and it bursts through the cell membranes. You burst the cell membrane, are you ever coming back? That cell's dead. You guys ever seen like climbers and things like that when they get frostbite? You ever seen when their fingers and things fall off? You have to go necrosis. What's happened is the fingers, the nose, the ears, they've frozen, the cells have burst, and they die, and then the flesh flakes off. Pretty gross. Right? That's what's happening. So that's at 0. 45, what happens at 45?
Protein.
The proteins in nature. What does that mean? Yeah, when have they stopped working? But the reason they stop working is they undergo a conformational shape change, which makes them non-functional. Or is it ever going back? Does the steak go back to cow? No. Yeah. Okay. So in this case, once you damage it to a certain degree, it's non-functional anymore. That's typically the upper and lower limit. Interestingly enough, however, there are certain organisms that can span those gaps. They can breach those boundaries. There are algae that can actually live in ice, right? There are fish that can exist right down to the point of where water is freezing, because they have all kinds of interesting physiological adaptations to do so. The fish has basically a form of natural antifreeze in its blood, which allows it to live there. You can get organisms that are basically found right next to boiling water. Lots of microbes can do this because they have the physiological mechanisms necessary to do it. So the way to deal with lethal extremes of temperature or water or any kind of physical or chemical attribute is via two processes: acclimation or adaptation. What's the difference? It's a time difference primarily. Which one's shorter? Acclimation is a short-term physiological or behavioral response to deal with some environmental pressure. What's adaptation?
You evolve to.
That's a long-term response, changes in morphology, behavior, or physiology at a genetic level, right? Changes in DNA that are encoded. So one is a long-term response and one is a short-term response. So if you live in some place like Arizona, like my mom used to live in Phoenix. Anybody from Phoenix? Good. You're in Phoenix, get the hell out of there. Terrible place. It's hot as hell. It's in the middle of some weird salt basin. I have no idea why anyone would ever build a city there, ever. And it's super hot. And I'd go there and I'd be like, oh my God, I'm dying. It's the hottest I've ever felt in my life. And she'd be like, oh, it's not hot at all. It's because they've acclimated. And they're doing subtle behavioral things that are different. For me, I just march around all day. In Phoenix, is that a good idea?
No.
Hell no. From 10 to 2, I'm inside in air conditioning someplace. I'm not marching around. So little subtle changes in behavior, what you're wearing, certain ways that you're carrying yourself, all kinds of different little things you can acclimate. There's lots of little physiological short-term responses you can do as well. You just don't feel as hot. Adaptation is very different. That's changing shapes, traits genetically through the process of natural selection, right, and mutation. Very, very different. So those are ways you can deal with it. Why do you have adaptations in the first place? The environment is nothing but throwing problems at you all the time. The environment is throwing problems. We're going to talk about this a lot in evolution. So each environment has a series of problems associated with it that you have to bash through as an organism to survive. If you can't deal with the problems, what happens to you? You die. It's that simple. And so adaptations are nothing but functional solutions to those problems. That's all they are. All right, so that's water. Let's talk about the impacts of water for organisms like ourselves. And so water is essential for life. Life is an aqueous process. No life as we know it without water. If I was to decompose you down to your most base form, you are 1 trillion bags of salt water with electrical stimuli running between them. And if I dried you up, you'd be nothing more than a little handful of dust. That's about it, as you're primarily water. Okay? And so controlling your water balance is critical for your existence. So organisms deal with it in different ways. So for instance, a shark. Is the fluid outside the shark more salty or less salty than its body fluids? It's more salty. So because of basic diffusion, where's the water going to go from the shark? Out. Right? This is going to be osmosis because it's flowing through a membrane that's differentially permeable, but the basic process is the same. So it flows out. So what's going to happen to the cells in the shark?
They're gonna shrink.
They're not going to burst, they're going to shrink. And if they get too small, there's not enough water, shark's going to die. It's literally going to dehydrate in an ocean full of water. But that's an easy problem for the shark to deal with. They're losing water, so what do?
They move solute out.
They open their mouth and they drink it. They're in water, right? You just drink more of it. So it's a super easy thing for a saltwater fish to do. It's hardly any problem at all. You're losing water, but you just drink it like that. It's a little bit different. Now, in that case, the fluid is hyperosmotic to the fish. But look at this fish down here. What kind of fish is that? The bottom.
Salmon.
That's a sockeye salmon. Is that a freshwater or saltwater fish? Well, that's kind of a trick question. At this stage, it's in the river, so it's what?
Freshwater.
That's a freshwater fish. So in this case, what's the fluid? Is it more or less salty than the organism itself, the fluid in the organism? It's less. So if that's the case, where's the water going to flow? In. So the cells on that fish are going to do what? They're going to swell up. And if you don't get rid of that water in the cells, what's going to happen to them? They're going to burst just like if they were frozen. And that's going to kill the organism. So how does a fish deal with it in fresh water then?
Pumps it out.
They pump it out. And how does a fish pump it out? They pee. That's exactly right. They pee. Freshwater fish are peeing machines. They pee all day and all night. A typical freshwater fish pees out 1/3 of its body weight per day. So if you're 150 pounds, that means you'd be peeing out 50 pounds of urine. You spend how much of the day in front of the toilet? Right? That's how pretty incredible when you think of that. So one is gaining water, one is losing water, but they have to balance that water properly for life. And then there's organisms like ourselves and terrestrial things. Now, most terrestrial organisms are continually losing water to the environment. More water in you or in the air?
In me?
Way more water in you than in the air. So where's the water in you going?
Out.
Out. And it's coming out through your skin and perspiration. Where else is it coming out?
You're breathing?
In your breathing. So to breathe, your lungs are saturated with water on that surface to keep them wet. So you have to maintain the wet or you can't breathe. And so what happens is every time you take a breath, you lose water. I'm going to lose way more water than you lose today. Why? Because I'm talking a hell of a lot. And I had a class before you. So when you see me dehydrated and passed out, throw water out. Okay? That's water. Yes?
So for saltwater fish, if they're losing, or if they're, so their environment's more salty, but then how is like consuming more salt
Because
water?
they have processes to be able to sift out some of the salt. There's all kinds of different physiological mechanisms like that. But the water is readily available for them to grab it just. Good question. All right, so we're losing water all the time. That's another couple places. 2 more places you'll lose it. Where else?
Urine?
Urination. You have to get rid of your nitrogenous waste products. When you have a protein, you cleave off the amino group, which is highly toxic. If you don't get rid of that, it poisons you. So the way you get rid of it is to attach it in water, which is soluble in water. You can lose it that way. So for us, what's the nitrogenous waste product?
Ammonia or urea?
That's urea, which is kind of like a modification of ammonia. You have to build it, it takes a little energy, but then you pee it out. For something like a reptile, they're even more water conservative than we are. They don't use urea, they use what?
Uric acid.
Uric acid, which is kind of a paste. It takes a lot of energy to do that, and they grow slower because of that. All right? So you lose it in defecation, and you lose it in urination. Right? Every time you defecate, you're going to lose water too. And we have all kinds of mechanisms physiologically to retain it. So when you take a look at an amniotic egg like this turtle's coming out of, that itself is a water retention device. That is taking the seed and encapsulating it in a shell so that you can dump it in some of the driest areas in the entire world. Frogs, amphibians are really prone to water loss because they have permeable skin. So they're really tied much more to wet environments. Something like this camel is not. So camels are found in some of the driest environments of the world. How do they get away with it? They've got a whole suite of adaptations to deal with it. That hump in particular is one of them. There's a misconception. Is there water in the hump? No. That's some weird falsehood. I don't even know where that comes from. But what is in the hood? The hump. Fat. So fat is a great what? Insulator. And it keeps the animal from getting hot. So that's what the whole back's covered in. When you look at the pads of the feet, they're super thick. They're filled with what? Fat. And that will allow, some of that ground can get up to 160 degrees. But they're walking on like it's no problem. A camel can basically refill 20% of its body weight in water within 10 minutes of drinking out of water bowl. If you did that, what would happen? You'd die from water poisoning. You'd explode, right? No problem for them. They can absorb heat through an entire daily period, retain it, and And then as soon as it starts radiating back out, then there's less energy required to do so. So you have all kinds of really cool adaptations for making a living in the environment. Are camels nice?
I don't know.
They're right up there with koalas for being pain in the asses. Mean, ornery, and slobbery. They spit too.
At least they're resourceful though.
They are very resourceful. And they're much smarter. All right, so that's the one. This is my bash the mammal day, I guess, right? All right, let's talk about light. Light varies place to place on the planet, varies in intensity, varies in its duration. It has direct influences on the quality of photosynthetic processes around the planet. So, the vast majority of energy that drives this planet comes from the sun in the form of solar radiation. There are other forms of energy that are available. What are they? There's chemical energy all around us. What else? And the microbial world uses that to great advantage. What else? Thermal. So there's geothermal heat. And so if you look at it, you find all kinds of geothermal venting and things like that. Those are all the forms of common energy on the planet. But most of it is run by light from the sun. Okay? So the intensity of quality of that's going to change, and that in turn will impact. Am I mic'd today? My voice is going on. Yeah, I'm mic'd up. I feel like it's smoking or something. Getting all raspy. All right. So development and behavior of organisms are directly dependent on what kind of plants you find. The plants you find and where they're found is directly related to the intensity, duration, and seasonality of the light. Soil structure is really critically important. So soil structure has different physical construct. It's dense. It can be porous. It can be nutrient loaded. It can be nutrient poor. So that will determine what kind of organisms can survive and live. The soil itself affects water chemistry because water flows against the soil. River water is directly dependent on the soil that the water flowed through to get into the river. The amount of salt is dependent on how much that is exposed to daylight, intensity of the heat. And in marine systems, this construct of the soil will determine the organisms you find, whether it's soft sands or rocks, will determine what kind of organisms you find in those particular habitats. So soil structure is really critical as well as a limitation to distribution of organisms. I don't find hard, encrusting, sessile organisms on soft bottom sands. They're limited in the distribution because of that material. Let's take a look at soil structure as a determinant of a limitation of distribution. And I want to talk about an interesting feature. So this is actually near Berkeley. And this is up kind of in Central Cal, near the Bay Area. And what you find up there is an interesting soil type called serpentine soil. Anybody from the Bay Area? There you go. So if you kind of go over the bay from San Francisco, and you're kind of on the East Bay side, you'll see this soil type. And it's called serpentine soil because it forms these outcroppings that have kind of a sinusoidal flow to them, and they look kind of snake-like. And also their coloration is real distinctive. So when you look at their color, they're kind of bluish to gray. Sometimes they'll appear a little green in coloration. And it's because of the minerals that you find in those rocks. So they're really high typically in nickels, very common, and also you see magnesium is quite commonly found in the rocks. Now, of interest is those soils have a high affinity for calcium. And calcium is bound tightly in this kind of soil in relation to the normal sandstone soils found around that one. So there's a kind of flower that you see growing on. Here's an example of it. When you look at this soil, the first thing you notice is hardly anything grows on it. Sandstone soil will be right next to it, and there'll be all kinds of different species of plants. But on the serpentine soil, there's hardly anything. Usually there's one single type of flower growing on it, and that's called the Goldfield flower. And it's a gold little flower that's sitting. Well, it grows there, but it's not in the sandstone soil, and other plants in the sandstone soil aren't in the serpentine soil. So what's your question? Why not? Right? Why? So in this case, we only find goldfields on the serpentine soil. Other types of plants are not. They're limited to the sandstone soil around. What's our tool for this test? Transplant experiment. So why are other plants found in the serpentine soil, and how does that serpentine soil affect their distribution? So we're going to run a transplant experiment. What's our hypothesis? Other plants can't live on the serpentine soil because it's toxic. Why might it be toxic?
Nutrient content?
Because it's full of what? Heavy metals, right? It's full of heavy metals. And also, it has another problem. Because it's heavily full of metals, it has low water retention capacity too. The other thing is, normal plants need calcium to drive their metabolism. So if I need calcium, what is this soil doing to the calcium? It's binding it, and I can't get at it if I'm a normal plant. That's a problem now. All right, so our experiment is we grab normal plants from the sandstone soils and we transplant them onto the serpentine. And I see if they do what? They survive and they reproduce. So I transplant them and they die. Can I say that that is because the abiotic constitution stink and that's what limits the distribution? Why can't I say that?
Control.
Because I didn't do what?
Control.
I didn't control for the transplant. How do I control for the transplant?
You dig it up and then you put it back.
I dig it up and I put it down. So next time we meet, I'm going to talk about what the ramifications